Homo Sapiens. Who are we? Where do we come from? In Chapter 20, we will continue our quest to find the beginnings of life on Earth, Darwin's spark of life. In Chapter 19, we looked at the nature of viruses and viroids, biological entities that occupy the shadowy borderlands that separate the prebiotic world from the biotic world. Somewhere in this metaphorical border region, between the viral entities and the sin ancestor, must lie Darwin's spark of life. Research points to the spark of life occurring on the Earth somewhere around the 4 billion year mark in Earth's 4.5 billion year lifespan, when our Earth was a very different and much more hostile place. But these hostile conditions on the early Earth were apparently the perfect incubator for Darwin's spark of life, which in itself can be viewed as some unique synergy of physics and chemistry. If we are going to have any chance of understanding Darwin's spark of life, we need to delve deeper into the complex biochemistry that underlies all of life. In Chapter 20, we are going to tackle this complex biochemistry with a look at a discovery that some scientists believe may represent a fourth domain of life, giant viruses. Let's begin. In 1992, Dr. Timothy Rowbotham was a microbiologist working for the Leeds Public Health Laboratory in England. While working for the Public Health Laboratory, Rowbotham had become fascinated with Legionnaire's disease and the Legionella bacterium that was the causative agent of the disease. An outbreak of pneumonia in the town of Bradford in West Yorkshire County in 1992 was a suspected Legionnaire's outbreak, which led Rowbotham to sample water from local cooling towers in the town, surveying for the Legionella bacteria. There had been a general consensus among scientists that the Legionella bacteria were free-living in fresh water, such as that found in cooling towers and other types of public plumbing. Rowbotham had discovered, however, that the Legionella bacteria were actually surviving in fresh water via a parasitic relationship with the amoebas that are commonly found in freshwater environments. Because the bacterial strains associated with Legionnaire's disease were difficult to grow in a normal laboratory culture, Rowbotham developed an amoebal co-culture methodology to isolate the bacterium within the amoeba. Amoebal co-culture is a laboratory method for growing and isolating amoebal parasitic bacteria by introducing a sample of the suspect water into a laboratory colony of amoebas. This methodology is based on the observation that certain types of bacteria, such as Legionella, are able to harbor in amoeba by avoiding digestion and are thus provided with a protective environment where they can survive and reproduce. Rowbotham used a colony of Acanthamoeba polyphasia as his bacterial culture or host medium to test the water samples he had collected from one of the Bradford cooling towers. Upon examining an amoeba specimen from his Bradford test colony under the microscope, Rowbotham noted a series of spherical or coccus-shaped bacteria. This seemed odd to Rowbotham as Legionella bacterium were rod-shaped bacterium. Rowbotham's initial thoughts were that he may have discovered a new bacterial infectious agent associated with Legionnaire's disease, and he thus named this new bacterium the Bradford coccus. Rowbotham's Bradford coccus bacterium passed the stain test for a gram-positive bacterial type, but other genetic testing failed to return any meaningful results which might help identify the phylogenetic nature of the bacterium. Rowbotham continued to try various methodologies over the next year to determine the nature of the bacterium, but his efforts met with failure. Still hoping to identify the Bradford coccus, Rowbotham passed the mystery samples on to Richard Birtles, a colleague also working at the Public Health Laboratory, who was finishing up his doctoral requirements for his PhD in microbiology. Birtles was among the first to begin applying a new molecular-based genetic testing methodology to profiling microbes, but he also met with failure in his attempts to extract any meaningful genetic material from the Bradford coccus specimen. With the attempts to genetically identify the Bradford coccus meeting with failure, Rowbotham froze the biological samples in hopes that future testing might reveal positive results. In 1998, Rowbotham's laboratory started closing down due to budget cuts, so he reached out to Didier Raoul, head of the Unité de Recherche, a renowned microbiology lab at the Aix-Marseille University in France, to see if he might be interested in the specimens. Raoul was very interested, and Richard Birtles, who who was doing postdoctoral work with Raoul at the Aix-Marseille University at the time, was able to transport Rowbotham samples to the Aix-Marseille laboratory, including the Bradford coccus specimen. Raoul and his colleagues at the university ran tests on the Rowbotham specimens, leading to the discovery of 4 new strains of Legionella bacteria. When Raoul and his fellow researchers at the Aix-Marseille laboratory turned to testing the Bradford coccus specimen, they began to encounter the same issues Rowbotham and Birtles had encountered in extracting any meaningful genetic material from the specimen. In early 2003, after continued failure of standard laboratory methods to identify the genetics of the bacterium, one of the researchers, Bernard Lascola, decided to examine the Bradford coccus under an electron microscope to determine if some novel structure was impeding their tests. Lascola, a bacteriologist, was surprised to see the Bradford coccus was clearly displaying a viral structure, but it was far larger than any virus ever seen. Lascola passed news of his discovery to Raoul, who sought the advice of Jean-Michel Claveret, a bioinformatics specialist at the Structural and Genomic Information Laboratory in Marseille. Claveret sequenced the virus's DNA, and what he found astounded him. The virus's genome was about 1.2 million base pairs in size, 10 times larger than the average virus and even larger than some bacteria. In the March 28, 2003 issue of Science magazine, the newly discovered giant virus, which had been dubbed the Mimi virus, was officially announced by Raoul and his team in an article titled, "A Giant Virus in Amoebae." The world of virology and microbiology was stunned as word spread of the new discovery. Let's now begin a detailed look at the Mimi virus and other strains of giant viruses and their impact on evolutionary biology and possibly Darwin's spark of life. What exactly are giant viruses and how do they differ from ordinary viruses? Let's proceed to answer this question. Since the discovery of the Mimi virus in 2003, researchers have probed numerous environments across the Earth, greatly expanding the known strains of giant viruses. Initially, our look will be restricted to a handful of the most prominent varieties of giant viruses, namely the Mimi virus, the Mama virus, the Megavirus, the Pandora virus, the Pithovirus, and the Tupon virus. In order to better understand the nature of giant viruses and what sets them apart from ordinary viruses, let's make a comparison between the Mimi virus and 4 mainstream or ordinary viral types. The 2 principal factors in differentiation will be physical size and genome size. For our mainstream or ordinary viruses, we will choose the rhinovirus, the influenza A virus, the measles or rubeola virus, and the smallpox or variola virus as our comparison examples. The rhinoviruses, viral agents of the common cold, are the predominant infectious viral agent in humans. Rhinoviruses are among the smallest of viruses at 30 nanometers in diameter, a nanometer being 1/1,000,000th of a meter. The influenza A virus is the most common variety of flu virus, with the other 2 varieties being type B and type C. Influenza or the flu is responsible for some 500,000 deaths worldwide each year. The influenza A virus is approximately 100 nanometers in diameter. The measles virus or rubeola virus is the viral agent responsible for the highly contagious disease, the measles. From 1855 to 2005, it is estimated that the measles killed some 200 million people worldwide. The measles virus is approximately 150 nanometers in diameter. The smallpox or variola virus is the viral agent that causes the dreaded smallpox disease. Smallpox is estimated to have killed between 300 to 500 million people across the earth during the 20th century alone. The smallpox virus is a brick-shaped virus roughly 350 by 270 nanometers in size. Our giant virus example, the Mimi virus, on the other hand, has a basic capsid diameter of about 400 nanometers, but protein filaments projecting from the surface of the capsid may add an additional 400 nanometers to the diameter, putting some observational estimates near 800 nanometers for the entire viral structure. Let me also note here that the largest known giant virus from the standpoint of physical size is the pithovirus, which measures in at 1500 nanometers by 500 nanometers, huge by viral standards. While the physical size difference between the giant viruses and ordinary viruses is significant, the most extraordinary and compelling aspect of ordinary virus versus giant virus is the size difference in the genomes, that is to say, their genetic makeup. If we compare the genome of the virus to the genomes of our four mainstream viral types, we can get a better sense of how the giant viruses really differ from our ordinary viral examples. Our first three mainstream viruses, the rhinovirus, the influenza A virus, and the rubeola virus, are all single-stranded RNA viral types. At the low end of our genomic scale, we find the rhinovirus with a genome consisting of about 8,500 nucleotides. The influenza A virus is next with a genome consisting of about 13,000 nucleotides. The rubeola virus weighs in with a genome of about 16,000 nucleotides. Our smallpox or variola virus has a genome built on a double-stranded DNA molecule consisting of about 186,000 base pairs, a base pair being the bonded pair of nucleotides that form the familiar cross-sections in the DNA spiral structure. In contrast, our giant virus specimen, the virus, which has a genome also built on a double-stranded DNA molecule, has a genome size of about 1,181,000 base pairs, which can be expressed more succinctly as 1,181 kilobase pairs, significantly larger than the genomes of our ordinary viral types. Moving on through our giant viral examples, from the standpoint of genome size, we would find the mama virus with its double-stranded DNA genome weighing in at 1,191 kilobase pairs. Next we have the megavirus with its double-stranded DNA genome coming in at 1,259 kilobase pairs. The Tupon virus, also with a double-stranded DNA genome, comes in at 1,500 kilobase pairs. The Pandora virus, with its double-stranded DNA genome, has the largest giant viral genome weighing in at about 2,500 kilobase pairs, or 2,500,000 base pairs. The pithovirus, with its double-stranded DNA genome, while the largest in physical size, comes in at the bottom of our genomic comparison of giant viruses at 610 kilobase pairs. The significance of these large viral genomes lies not only in the size, but in the genes that encode for various biological functions previously thought to be found only in cellular organisms of the life domains bacteria, archaea, and eukarya. Because of this unique genomic encoding found in giant viruses, some researchers have proposed that giant viruses represent a fourth domain of life on Earth, standing beside the domains of bacteria, archaea, and eukarya. To paraphrase from a scientific article in Frontiers in Microbiology, megavirals, the proposed order of classification for giant viruses, has increasingly demonstrated that it stands in the tree of life beside bacteria, archaea, and eukarya, and the megavirus ancestor is suspected to be as ancient as the ancestor of cellular organisms. While this concept of a fourth domain of life for megavirals is still in debate, there can be no doubt that giant viruses represent some insight into the prebiotic-biotic nexus of life on Earth. Let's now take a look at the deeper genetic mysteries of giant viruses or megavirals and what significance they may have for the advent and evolution of life on Earth. Let's begin our look at the genetic mysteries of giant viruses by introducing the monophyletic clade that contains the giant viruses. Monophyletic meaning descendants of a common ancestor. This monophyletic clade of complex double-stranded DNA viruses goes by the name of nucleocytoplasmic large DNA viruses, which often appears abbreviated NCLDV in scientific articles. The nucleocytoplasmic designation arises out of the observation that most of the viruses in this viral order can replicate in either the nucleus or cytoplasm of the eukaryotic host organism. The conundrum of this group of viruses lies in the fact that their genomes contain genes involved in DNA repair, DNA replication, transcription and translation, biological functions thought to lie solely in the domain of cellular organisms. Let's take a closer look at a few varieties of giant viruses and see what makes their genetic composition so unusual and compelling. The Pandora virus was discovered in 2013 by the husband and wife team of Jean-Michel Claverie and Chantal Abergel, researchers involved in the study of giant viruses. Two species of Pandora virus, the Pandora virus salinus, found in seawater off the coast of Chile, and the Pandora virus dulcis, found in a shallow freshwater pond in Australia, testify to the far-flung range of the Pandora virus. The Pandora virus salinus has the largest genome among the known varieties of giant viruses, with about 2,500,000 base pairs comprising its double-stranded DNA molecule. One of the most intriguing aspects of the Pandora virus genome is that about 93% of its genes are found in no other microbe on Earth, making its genetic makeup exceedingly unique. The replication cycle of the Pandora virus is also atypical. The Pandora virus virions, the virion being a single viral entity, are taken into the host amoeba via phagocytic vacuoles, whereupon the virions fuse with the vacuole membrane and via a pore on the virion apex release their genetic material into the cell's cytoplasm. Within 2 to 4 hours, the amoeba's nucleus is reorganized, and 8 to 10 hours later, new Pandora virus particles appear where the amoeba's nucleus once existed. This ability to hack the amoeba's nucleus and hijack its genetic machinery for the reproduction of Pandora virus virions goes to the heart of the genetic nature of life, which at its most basic level comes back to DNA or RNA. Life is about reproduction and survival, as seen on display in this co-evolving and ancient dance between viral and cellular entities, in our case, the Pandora virus and the amoeba. In past chapters, we have looked at three fundamental aspects of the living organisms of the domains bacteria, archaea, and eukarya. 1. Information handling purpose to fateful reproduction via genetics, namely DNA. 2. Harnessing energy via metabolism. And 3. A homeostatic environment to house the life processes via a cell membrane. Regular viruses exist and reproduce at a very minimal level, relying solely on genetics, but dependent on the genetic machinery of living cells for reproduction, the virus itself having no metabolism nor cell membrane. Giant viruses have a more robust set of genetic tools, and while still relying on living cells for reproduction, seem to hint at a genetic prowess closer to living cells, harboring genes seen in cellular processes, but still no metabolism or cell membrane. Does this greater genetic prowess of giant viruses, such as the Pandora virus, hint at megavirals being a stripped-down version of some ancient ancestor from a hypothesized fourth domain of life, or is their genetic prowess based on genes borrowed from the eukaryotic life forms upon which they prey? This is the question that lies at the crux of the nature of giant viruses, and a question that is still in debate. Next, we will take a look at the Tupon virus. The Tupon virus comes in two varieties, the Tupon virus Deep Ocean and the Tupon virus Soda Lake, which were discovered in Brazil by a team of researchers led by Bernard Lascola. The Tupon virus Soda Lake has a genome size of about 1,439 kilobase pairs, while the Tupon virus Deep Ocean has a genome size of about 1,516 kilobase pairs. The most intriguing aspect of the Tupon virus is that it has the largest set of protein-coding genes found in any known megaviral genome, being able to produce up to 1,425 proteins in the case of the Tupon virus Deep Ocean. Among the known giant viruses, the Tupon virus possesses genes uniquely capable of translating all 20 standard amino acids being the fundamental building blocks of proteins. The Tupon virus, like the Pandora virus, also contains a significant number of genes found in no other life forms on Earth, with about 30% of the Tupon virus's genes being unique to itself. The Tupon virus, with its presentation of the largest protein coding or translation apparatus in the known viral sphere, seems to straddle the canonical frontier between the cellular world and the viral sphere, adding further intrigue to the mysterious nature of giant viruses. Lastly, let's take a look at the mama virus and a surprising revelation associated with the discovery of the mama virus. The mama virus was discovered in 2008 in amoebae collected from a cooling tower in Paris by a research team led by DJ Raoul. The mama virus is closely related to the Mimi virus, but larger in size than the Mimi virus. While researchers were studying the mama virus specimens under an electron microscope, they made an intriguing discovery. They observed a smaller virus attached to the mama virus, which they named Sputnik, after the original Soviet space satellite. The name being a pun of sorts, as smaller viruses closely associated with larger viruses are termed satellite viruses. The smaller virus Sputnik was found to have a circular double-stranded DNA genome containing only about 18 kilo base pairs, far smaller than the 1,191 kilo base pairs of the mama virus genome. The most interesting aspect of the Sputnik virus was that it was found to be a parasite of the mama virus, and as such was dubbed the first known viral phage, meaning a parasite of viruses. When a mama virus infects an amoeba, it uses its large array of genes to set up a viral factory within the amoeba for the purpose of self-replication at the expense of the host amoeba. It was observed that the Sputnik virus was able to infect the viral factories of the mama virus, hijacking their genetic machinery for the purpose of replicating the Sputnik virus itself. This Sputnik infection resulted in fewer mama virus particles being produced, and those that were produced were often deformed and less capable of future infection. This discovery of a giant virus, the mama virus, falling ill via infection by another virus, the Sputnik virus, fueled speculation as to whether giant viruses could be considered alive. To quote Jean-Michel Claverie, "There's no doubt this is a living organism. The fact that it can get sick makes it more alive." The debate over the nature and status of giant viruses continues, but recent metagenomic research has offered up new and far-ranging implications for giant viral studies as well as biological diversity in general. To digress for a second, metagenomics is the study of the genomic content of a complex mixture of microorganisms such as might be found in a sample of seawater or any general environmental sampling. Under ordinary protocols, DNA or RNA would be collected and sequenced from a given specimen or organism. Metagenomic protocols sequence genetic material from microbial communities as a whole that populate samples such as those taken from ocean water, soil, or even the microenvironment of the human gut. Thus, via metagenomic sampling and sequencing, it has been possible to discover the hidden diversity of microbial life, including giant viruses, in various environments without the knowledge or identification of the specific organisms themselves. These comprehensive metagenomic techniques have been made possible by advances in high-throughput sequencing, along with an increase in computational power that is critical towards parsing the huge volumes of generated data. The metagenomic studies of ocean waters facilitated by these technological advances have allowed researchers to discover an abundance of genetic sequences closely related to giant viruses, leading to a suspicion that giant viruses are a common parasite of ocean plankton. Metagenomic studies of ocean water further revealed that 70% of viral genes identified in the ocean surveys have never been seen before. This led researchers to speculate that giant viruses and possibly viral phages such as the Sputnik virus may play some role in regulating the growth and death of ocean plankton, thus having a major impact on marine nutrient cycles and climate. To paraphrase Curtis Suttle, a researcher in marine viruses from the University of British Columbia, I think ultimately we will find a huge number of novel viruses in the ocean and other places. these viruses could be a major player in global systems. In light of these metagenomic studies, the role that giant viruses play in our planet-wide ecology and the implications of that role are still being discovered. Whatever insight these studies may provide, it seems certain that giant viruses and viruses as a whole demonstrate the fundamental role DNA and RNA play in shaping the nature of our planet. Bacteria, archaea, and eukarya, the latter of which includes all of the more complex life forms on Earth, including ourselves, are DNA-based life forms. Viruses display an innate ability to manipulate DNA across all three domains of life, impacting Earth's biosphere on a global scale with far-reaching implications for natural selection and evolution. The planetary and geological entity that is our Earth is so intrinsically entangled with life in all of its forms as to be unrecognizable without that DNA-based life. And what is DNA itself but a molecular construct of atoms of hydrogen, oxygen, nitrogen, carbon, and phosphorus cobbled together by the chemical processes of nature to form nucleotides of adenine, thymine, guanine, and cytosine, which in endless combinations are responsible for the countless genomes that encode all life on Earth. When I started these last 5 chapters, back in chapter 16, my goal was to seek the answer to how life began on Earth. We have delved into some arcane biochemistry and looked at possible avenues for the advent of life here on our planet, while reflecting on the possibility that life may arise wherever conditions are ripe in our universe. The fact that life did arise on our planet is a given. RNA and DNA lie at the center of it all, a tangled tale with viroids, viruses, and giant viruses on the prebiotic side, and bacteria, archaea, and eukarya on the biotic side, and who knows, in the middle possibly, some transitional life forms now long extinct. So while the exact nature of Darwin's spark of life still remains elusive, the answer almost certainly lies in the realm of chemistry and physics. In Jurassic Park, Dr. Ian Malcolm noted that life will find a way. I believe in our world that science will find a way, and in some not too distant future, we will discover the nature of Darwin's spark of life. In Chapter 21, my goal is to return to the examination of human evolution with a look at the most recent findings in that field, as the dynamic world of science and research is always remolding our knowledge of who we are and where we come from.